Okay, so last class we left off talking about the pancreas, and we got up as far as the pancreatic enzymes, and that's what we're going to start off talking with today. So you can see on the slide here now that we're going to talk about a bunch of the enzymes released into the pancreatic juice, secreted by those pancreatic Acini cells, part of the exocrine function of the pancreas. And we can see that there's a pancreatic amylase, and we know what an amylase does, breaks down starch. We see here there's a pancreatic lipase, and we know what lipases do, right? They'll break down complex lipids. And then this is the first enzyme that we're seeing in the digestive system, a nuclease, and this is going to help us break down nucleic acids. It's going to break us down into those monomers of nucleic acids, and it's going to help us recycle our DNA and RNA. We're also going to have proteolytic enzymes. Now, if you're not sure what that word means, let's have a closer look at it. Proteo is referring to proteins, and lytic means to split, and that's exactly what proteolytic enzymes do. They break apart proteins. Now, we've got two classes of proteolytic enzymes. We've got the first one are the proteases. Now, proteases are going to be enzymes that deal with large proteins. And from a protease, we're going to take a large protein, and we're going to get the enzyme cut up in a little bit, and we're going to end up with smaller peptide chains. So still long chains of amino acids, but we're not going to have them in complex protein form anymore. From those peptide chains, then we're going to get a second class of enzymes called peptidases, and these peptidases are going to work on those peptide chains and break them into amino acids, which are much easier for us to absorb and then easy for our body to use after we absorb them. Now, the pancreatic proteolytic enzymes are all secreted as inactive proenzymes, okay? They only become activated when they reach the small intestine. And again, this is for a safety thing. We talked about when we talked about pepsin, how it's not a good idea to have a proteolytic enzyme secreted directly by the cell because it can actually digest our own tissue. So, when we look at the proteases from the pancreas, we're going to talk about 4 different enzymes, and like I said, they're all secreted as proenzymes. So the proenzymes are going to be trypsinogen, chymotrypsinogen, procarboxypeptidase, and proelastase. So all of those compounds are going to be secreted by the pancreas. They're going to follow down the pancreatic duct, enter into the small intestine, and in the small intestine they're going to come in contact, trypsinogen anyways, is going to come in contact with enterokinase. Now, enterokinase is an enzyme that's produced by the brush border cells of the small intestine. Once trypsinogen comes in contact with enterokinase, it becomes trypsin, and now this is our active proteolytic enzyme. Once trypsin is activated, then we have trypsin going to activate chymotrypsinogen, which becomes chymotrypsin. Trypsin will also activate procarboxypeptidase, which turns it into carboxypeptidase, and it'll also activate proelastase, which will turn it into proelastase. So we end up with 4 proenzymes. Once the first one, trypsinogen, is activated by enterokinase to become trypsin, then trypsin will activate the other 3 proenzymes to produce the 3 final enzyme products: chymotrypsin, carboxypeptidase, and elastase. And all of those are going to be proteolytic enzymes. Alright, so we've talked about the enzymes now. We've talked about what chemical digestions can happen in the small intestine. Let's talk a little bit about a typical meal. So when you eat a typical meal, it's going to contain a lot of different things: carbohydrates, proteins, lipids. It's going to have water. There's going to be electrolytes. There's going to be vitamins. There's going to be nucleic acids. There's going to be a lot of things that are in the food that we eat. The first thing, of course, that we need to do is we need to break those molecules, those larger molecules, down. We won't be able to absorb them as they are. And this is where we're going to get our physical digestion, so mashing them up, increasing the surface area. And it's also where we're going to get our chemical digestion, coming into contact with enzymes so that we can break chemical bonds. So that's going to fall under the category of digestion. And then once we break those bonds and we've got those things into smaller molecules, then we can absorb them. Some of the smaller molecules that we eat with our food, water, electrolytes, vitamins, we have specialized sources, cells. We also have those molecules are small enough anyways that they basically just get absorbed without any processing. It doesn't cost us any energy to absorb water, electrolytes, and vitamins. Alright, let's have a quick look at the carbohydrates. Now when we eat a carbohydrate, we're talking about usually large complex carbohydrates like starch, and we have these amylase enzymes, salivary amylase in the mouth, the pancreatic enzyme secreted in the small intestine, and those are going to break down large monosaccharide chains into smaller molecules that we can process. So we've got our polysaccharides, which means many sugars, so these polysaccharide chains can be really, really long, and they come in contact with salivary amylase or pancreatic amylase, and we get them broken down into smaller molecules, disaccharides, trisaccharides, which means we're only going to have 2 or 3 of these saccharide molecules linked together. The next step is to make those 2 and 3 or di and trisaccharides a little bit smaller and easier for us to use. And then this is going to happen at the brush border. Okay, so these disaccharides and trisaccharides are going to be in the lumen of the intestine. They're going to come up against the brush border, and they're going to come in contact with these enzymes like maltase, sucrase, and lactase, and that's going to take those disaccharides and trisaccharides and finally break them down into a monosaccharide like glucose. Glucose can easily be absorbed across the lining of the intestine, and then once we get glucose into our once we've absorbed glucose, it can enter directly into our metabolic pathway, which you guys know starts off with glycolysis, moves into the citric acid cycle through the electron transport chain, and we end up with 36 molecules of ATP. Now, proteins, it's going to follow the same structure, except we're going to be talking about different enzymes. So we're going to start digesting protein in the stomach when we have pepsin. Then we're going to move into the main protease in the small intestine, which is trypsin. So we're going to start off with these big, long, globular-shaped proteins that are long chains of amino acids. They're going to come into contact with pepsin and trypsin. Those are proteases, and they're going to break them into smaller peptides, strings of amino acids. Once we get into the small intestine, we'll have those enzymes that are peptidases, and they're going to cut those peptides down into amino acids, which we can easily absorb and then use. So the final digestion of the protein is going to take place again at the brush border. That's where we're going to find those peptidases, and those peptidases are going to take those peptides and break them down into amino acids, which we can readily absorb. Now, lipids are a little bit of a different story. They have to be packaged a slightly different way. We do have the major enzymes that will break down lipids. We've got the lingual lipase secreted by the tongue, the pancreatic lipase by the pancreas, and the gastric lipase, which we see in the stomach. Now, what those enzymes are going to do is they're going to take our triglyceride down here. So here's our molecule of glycerol, those blue chains, and then we get 3 fatty acid chains, 1, 2, 3, and those lipases are going to split that into a monoglyceride, which is this guy right here, and then long chain fatty acids, which are red circles right here. Now, breaking this triglyceride down into monoglyceride and fatty acid chains is going to make it a little bit easier for us to absorb. No. When it comes down to absorption, we need to have a look at the structure of the villi that are in the small intestine. Okay, so just to give you perspective, here is one villi, and the brush border that we've been talking about is going to be the area just right on the outside out here, going to be covered in microvilli, and that's where those brush border enzymes are going to be. But let's look at a bit bigger scale. Inside each villi, we're going to have the red blood vessel here, which is going to be an arteriole, and then we're going to have the blue blood vessel on this side, which is going to be the venule. So you guys hopefully will recognize this, where a vein and an artery or arteriole meet, as a capillary. Okay, and capillaries are specialized for absorbing things into the blood system, into the circulatory system, and for getting waste products out of the circulatory system. So remember that each villi has a capillary inside of it, and it also has this pink blind end sac that you can see in the middle, and that's going to be called a lacteal, and we'll talk more about that in the coming slides. So keep that structure in mind. Now, by the time blood gets, or by the blood, when blood is carried to the villi, it comes in the arteries, of course, and those blood is going to be low in nutrients. Okay, it's going to be coming from the lungs, from the heart, and it's going to have high oxygen, but it's going to have low nutrients. Now, when it comes close to the lumen of the small intestine, there's going to be a lot of nutrients in there because we just did all that digestion. So what's going to happen is we're going to have diffusion along concentration gradients from an area of high concentration to low. So we're going to see nutrients move from the lumen of the intestine across the intestinal cell membranes, across the epithelial cells, and into the arteries or the arterioles. Once they get into there, they move into the blood, they get transferred into the circulatory system, they get collected into a vein and carried away from the digestive system. And that's basically how we absorb those nutrients that we get as we're digesting our food. Now, that's important when we're talking about absorption into the circulatory system. Most of the things, when we absorb them, will go into the blood and they'll be carried away from the digestive system. One thing that we can't do is we cannot efficiently pack lipids. And that brings us to the story of the lacteal that's in the middle of the villi. The lacteal is a part of the lymphatic system. So lacteals will absorb lipids that are too big to get into blood capillaries. Okay, so they'll take the lipids that move across the membrane and they'll carry them along the lymphatic vessels, and eventually those lymphatic vessels will empty into the venous or the blood circulation, and that's how lipids enter our circulation to be processed and to be brought around the body. Now, lipids are a bit tricky because they're not water soluble. So here's where the bile salts come in, and we've talked really briefly about bile salts earlier. Our bile salts will emulsify lipids into tiny droplets called micelles. Now, micelles are little tiny, think of them as little circles, and I'll show you a picture on a slide. If you're ever washing dishes and if you had a meal that's had a lot of fat and there's little tiny grease puddles on the surface of the water, if you add a drop of dish liquid, that dish liquid will act as bile salts, and it'll help to emulsify the fat that's on the water and in the water that you're washing your dishes. You'll notice that it turns into little tiny circles, and those are the micelle components that are changing the shape or changing the arrangement of the lipids. Anyways, these micelle components will diffuse easily across the membranes, so it makes it easier for us to absorb. But what happens is the components of the lipids, they'll get resynthesized, they'll change back into larger molecules once they get into the intestinal cells. So once they do that, they're packaged into these molecules called chylomicrons, and then those chylomicrons will enter into the lacteal by endocytosis. So here's a look at it here. Here's our micelle. So our micelle takes those lipid bits and pieces. We've got the white squiggly lines are going to be the bile salts. So the bile salts are going to organize those bits and pieces of lipids, and they're going to form a circle or like a little ball, which is the same thing you see in your dishwater. Once it comes up against the brush border, the edge of the epithelial cells, you're going to have those monoglycerides and those fatty acids move across the membrane into the cells. Once those bits and pieces of lipids get inside the cells, you'll see they'll go from being a monoglyceride, and they'll reform themselves into triglycerides. Once they do that, then they can be absorbed into the lacteal and they can get into the blood circulation. Now, where do all these nutrients go when we absorb them into the blood? Well, they're collected. They have a special blood circulation, and they get passed into the liver. And when we come back in the next video, we'll pick it up and we'll talk about the important function of the liver.